Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on double helical structure of DNA. So it is also called as Watson and Crick model of DNA. So DNA is a largest biomolecule that consists genetic information. So the genetic information present on DNA which is enclosed in the nucleus of the cell. Okay. So if you see the double helical structure of DNA, so it consists two polynucleotide chains. They arranged anti-parallel to each other. So DNA double helix consists two anti-parallel DNA strands. So each strand is made up of nucleotides. That's why DNA is a polynucleotide chain. So in the earlier classes we have already discussed about the nucleotides. If you want clear explanation about nucleotides, you can see the previous video. Okay. So DNA is a polynucleotide chain. It consists deoxyribonucleotides. Why? Because DNA consists deoxyribose sugar. That's why the nucleotides are deoxyribonucleotides. Okay. Deoxyribonucleotides and their ability to form a polynucleotide chain was first observed by the scientist Levin in the year 1931. Erwin Chargaff he discovered that DNA of any cell of the organisms, it contains 1:1 ratio of purines and pyrimidines. Okay. So according to Erwin Chargaff, he states that the DNA from any cell of any organism that contain 1:1 ratio of purines and pyrimidines. So that means the number of purine bases is equals to the number of pyrimidine bases. So you know purines are A plus G. The number of purine bases is equals to the number of pyrimidine bases. So this rule was proposed by the scientist Erwin Chargaff. Not only that, he also proposed the quantity of A is always equals to the quantity of T. The quantity of G is always equals to the quantity of C. This rule is popularly known as Chargaff rule of molar equivalence. According to Chargaff, A is always paired with T. That means adenine is always paired with thymine and guanine is always paired with cytosine. This is called a Chargaff rule of molar equivalence. So initially there are two scientists. They are Maurice Wilkins, Rosalind Franklin. So actually they worked on DNA. So they wanted to know the structure of DNA. They worked on DNA before Watson and Crick. So they wanted to know the arrangement of atoms present on DNA. So they performed a technique nothing but X-ray crystallography technique. By using this technique, they got fine X-ray photographs of DNA. So the photographs revealed the diameter of DNA, the length of the helix, and the distance between two base pairs also. Okay, so they revealed those important features of DNA double helix. Okay, so they revealed that the diameter of DNA is 22 angstroms, the length of the helix is 34 angstroms, and the distance between two base pairs is 3.4 angstroms. These facts facilitates Watson and Crick to give the exact structure of DNA double helix. So, the model proposed by Watson and Crick was widely accepted by many scientists. Now also we are accepting the Watson and Crick model of DNA. So, let's see the main features of Watson and Crick model of DNA. The DNA molecule consists two polynucleotide chains. So these are the two polynucleotide chains. Okay, it consists two polynucleotide chains. Each strand consists many nucleotides. So in between nucleotides there are phosphodiester bonds. Okay, so the backbone of DNA is nothing but phosphodiester bond. So the nucleotides are joined together by phosphodiester bonds. So phosphodiester bond is nothing but 5' carbon, oxygen, phosphate group, again oxygen, again 3' carbon. Okay, so this is established in between two sugar molecules. So we have already discussed about this in the previous video. So next one is the two strands of DNA, they are arranged anti-parallel to each other. Okay, so anti-parallel is nothing but one strand is consisting 3' to 5' direction. The opposite strand is consisting 5' to 3' direction. So that is the anti-parallel nature. One strand is consisting 3' to 5' direction and another strand opposite to this that consisting 5' to 3' direction or polarity. So that is called anti-parallel nature of DNA. So the anti-parallel nature of DNA, which is essential for the formation of hydrogen bonds in between nitrogen bases present in the two strands. So the anti-parallel nature that facilitates the formation of hydrogen bonds. So here you can see the nucleotides of one strand, they connected to the another strand by hydrogen bonds. So if you see, this is the one strand, this is the another strand, it consists nucleotides. It is a polymer of nucleotides. They are held together with the help of hydrogen bonds. Okay, so the hydrogen bonds are established in between purines and pyrimidines. One purine is always binds with pyrimidine. So for example, A always binds with the pyrimidine T with two hydrogen bonds, vice versa. Vice versa nothing but T always binds with A. Thymine is always binds with adenine. G always binds with C with three hydrogen bonds, vice versa. That means C always binds with G with three hydrogen bonds. Okay. So, in between two strands of DNA, there are hydrogen bonds. They held two strands together. So, this kind of base pairing in between purines and pyrimidines is nothing but complementary base pairing. actually the hydrogen bonds are relatively weaker bonds. Even though the DNA helix consists so many hydrogen bonds. Okay. So many hydrogen bonds, they are established in between two strands. So that held the two strands together. That gives strongness to the DNA. So if you wanted to separate the two strands of DNA, so heating can separate the two strands, and an alkali treatment that separates the double helical structure. So it gives two strands, two separate strands. Okay. When you treat the DNA with heating or with an alkali, it gives separation of two strands. Then two strands get separated. So this separation is called as denaturation. The separation of DNA by heating or alkali treatment is called as denaturation. Okay. So the two strands of DNA helix, they coiled together in a right-handed helix. Thus, it forming the DNA duplex or DNA double helix. So, the diameter of the helix is nothing but 20 angstroms. Okay. So, actually it consists a major groove and a minor groove. So, this is again major groove. Actually, this is called one helix. It consists one major groove and one minor groove. Okay. So this is called a helix, single helix. Okay. So it consists the length of 34 angstroms. Each helix consists 10 base pairs. The distance between two base pairs, nothing but the distance between these two base pairs, G and A, is equal to 3.4. Because each helix having 10 base pairs. Okay, 34 by 10 is equal to 3.4 angstroms distance present in between two base pairs. Okay, that means each helix is having 10 base pairs. So it consists a right-handed helix. Each turn consists a major groove and a minor groove. Actually, proteins are interacted in these grooves. You know that the DNA molecule can duplicate themselves. DNA can self-reproduce themselves. So the DNA double helix can produce two DNA daughter strands. So that is nothing but replication. So here you can see the replication of DNA. So two strands get separated during the replication. Each strand can produce new strand. So this is nothing but DNA replication. Okay, in this way, in the new strands, one strand is parental strand and that acts as a template that synthesizes the new strand. So there the complementary bases arranged according to the template strand. Complementary bases will come, they join together to form a new strand. In this way, single DNA molecule can produce two daughter molecules. That is nothing but replication of DNA. Okay students, this is about the Watson and Crick model of DNA. Thanks for watching.